Hello everyone. Today I am going to demonstrate a very important fundamental topic of cell biology, which is cell size and cell theory. So first we talk about cell size and now move towards cell theory. So first, it's the introduction to cells. As I already discussed in previous video about that cell is the fundamental unit of life. It is the smallest unit of organisms that we can consider alive. So the first point is cells are extremely small. So cells are so small that we have to use different types of microscopes to observe the cells. If we want to study the cells and their structures, their organelles, we need some type of instruments to observe it. So this instrument is actually a microscope. So some cells and larger cells can be seen with naked eye, but many smaller size cells like microscopic even microorganism cells, you can't see them with naked eye. Even multicell organisms made up of cells, we cannot observe them with naked eye. So microscopes allow scientists to study cell and their structures. So typically different organisms have different cell sizes. Here, we can see that bacteria, typically the size of bacteria is varying from zero point 0.5 to 10 micrometer. Plants and animal cells that are multicellular organisms, so the size of their cells are also larger, and it is about 50 to 150 micrometer. And here are some examples of unicellular organisms, like Chlamydomonas. The size of Chlamydomonas is 30 micrometer, whereas amoeba, whose cell size is much larger than Chlamydomonas, which is about 400 micrometer, while the last is Escherichia coli, and it is a bacterium. As I already tell you that the cell size of bacteria is vary from zero point 0.5 to 10 micrometer. So E. coli has 2 micrometer cell. So these are some examples of different organisms having different cell sizes. So we need some unit to measure the cells. That when we observe the cells under the microscope, we have to measure in. We need some type of units to measure them. So typically we know that meter is a unit of distance or length. So but meter and it is a SI unit, means standard international unit. But it is a much larger unit to measure the cell. So we need some smaller units to measure the different dimensions of cells. So we will convert into a smaller unit. Here is the conversion. We can see that 1 meter is actually equal to 1000 millimeters. 1000 millimeter. And in 1 millimeter is actually equal to 1000 micrometers. And we can write micro like this way, micrometers. So when 1 micrometer is actually is equal to 1000 nanometers, and means 10 raised to power minus 9. So we have to measure the cell sizes in micrometers. And then now the question arises, why we use micrometers to measure the different cell dimensions. Here I add concept question, which is very important for exam point of view. Why are micrometers used to measure cells instead of millimeters? So the answer is cells are extremely small. Therefore, micrometers provide a more convenient and accurate unit for measuring cell dimension. So that's why we use micrometer instead of meter or millimeter, because these units are relatively larger than micrometers. So here is a calculation question. How we can convert how many cells of diameter of 100 micrometer can fit side by side in 1 millimeter cell? Means if we have a cell of 1 millimeter, how many cells of 100 micrometer can fit in it? So this question has different steps. The first step is when we have to solve such type of question, we need to convert the units of both values. Means both values have same units. So we already talk. Then I already tell you that we use micrometer to measure the cell. So we will convert 1 millimeter into micrometer. So here 1 millimeter is equal to 1000 micrometer. So here the next step is we will divide 1000 micrometer into 100 micrometer. 1000 micrometer into 100 micrometer. And the answer is 10 cells. Means we can fit 10 cells of 100 micrometer into 1 millimeter. So this is an answer. Okay. This is all about cell size. Now moving ahead, I am going to discuss the cell theory. How early scientists study the cell, their structures, and using different microscopes. It is a very interesting topic and very important topic also for exams. So, actually, the study of cells began more than 300 years ago, after the invention of different microscopes. And one of the first scientists to observe cells was Robert Hooke in 1662. And what he observed, he used this microscope to observe uh, cork of plant cells, cork of plant. And actually, he observed dead cells of plant by using microscope. So, actually, Robert Hooke observed a thin slice of cork using a microscope. He saw many teeny box-like structures that looked like small rooms. So, because they resembled the small rooms used by monks, which were called cells, he named these structures cells. So, here important point for students that Hooke observed dead plant cells in cork. So, the next scientist, Antony van Leeuwenhoek. And actually, Antony van Leeuwenhoek built a very powerful microscope, which can magnify the object to 40 times. And by using this microscope, he observed microorganisms like bacteria and protozoa and different blood cells and sperm cells. So it was a great achievement to observe the microorganisms by using microscope. And another important scientist was Robert Brown. In 1831, Robert Brown discovered a nucleus, an important structure inside the cell. It is a very important organelle inside the cell. So next is the scientists. As microscopes improve, scientists began to understand that cells are the fundamental units of life. So he developed a cell theory. So firstly, two scientists, Sheldon and Schwann, proposed that all cells are the basic unit of life. Means all living organisms are made up of cells. In 1838 and 1839. Later, Rudolf Virchow added another key idea. He stated that new cells arise from pre-existing cells. So through cell division means if we have a cell, it will be divided, and by this division, two new cells will be formed. This is the key idea. A cell arises from pre-existing cells. So in 1862, Louis Pasteur disapproved the idea of spontaneous generation. And what is spontaneous generation? Actually, some scientists thought that living microorganisms or microorganisms come from non living matter, but like it came into existence spontaneously. But Louis Pasteur disproved this idea by using an experiment. Now I'm going to describe this experiment. What Louis Pasteur actually done. So Louis Pasteur used a swan neck flask experiment. Swan neck. This is a swan neck flask experiment. So first what he did, he sterilized neutron broth, and this is a neutron broth. Neutron broth means he used the liquid media which contain neutrons, and it is used to growth the it is necessary for the growth of bacteria. Like neutrons is a food for microorganisms to grow. First he sterilized it by heating. Like all type of bacteria or other microorganisms were killed. So then in the second step, what he did, he actually allowed the air to enter. He allowed the air to enter the growth, but he don't air was allowed to enter, but microorganisms cannot enter in this growth. Why? Because the flask, the neck flask was very narrow that microorganisms cannot enter here. So what he observed? He observed that no microbial growth occurred, and it proved that microorganisms come from existing microbes in the air, not from spontaneous generation. Means if microorganisms enter in the growth, that new microorganisms were formed, and he observed the growth of microorganisms on another bacteria. So by using this experiment, he observed that the living microorganisms, there is not any type of idea of spontaneous generation. He actually disapproved this. Okay, this is the contribution of Louis Pasteur. Okay, from these discoveries, scientists developed the concept known as cell theory. Cell theory actually states the three very important concept, three very important ideas. And what are these? First, the cells are the building blocks of living matter. Means all living organisms are made up of cells. The second one is cells are the smallest unit of life. Means it is the basic unit of life. And the third one, cells arise from pre-existing cells. Means cells like cells are formed from by using cell division. Means when this cell will be divided and new cells will be formed from these pre-existing cells. Okay. Modern biology has added two more idea to this theory. Means cell also contains genetic information, usually in the form of DNA, which control the growth and development of an organism. Cells are also site of metabolism, mean all chemical reactions necessary for life occur inside the cell. So this is the cell theory. Hope you will understand all the points. First, I had discussed cell size, microscopic units, and the contribution of different scientists in cell study, and the last cell theory. So today this is my topic. This was my topic. I hope you will enjoy it. Thank you very much.